Hello students, I am Dr. Harkamal Singh. Today we are going to study Wuchereria bancrofti. Wuchereria bancrofti is a human parasitic roundworm that is the major cause of lymphatic filariasis. Wuchereria bancrofti was first demonstrated in 1876. Its first adult female was demonstrated by Bancroft, hence its name bancrofti. Its first male was described in 1888. Its first intermediate host Culex mosquito was described in 1878. Geographical distribution. Wuchereria bancrofti is very widespread and important human parasite in the warm countries because warm environment supports the growth of insects. So Culex mosquitoes are very high in number in the warm countries. That's why Wuchereria bancrofti is also very prevalent in the warm countries. It is present in Africa, in Asia, in Arabia, India, Malaya, north to China. All the countries, warm countries, are affected by the Wuchereria bancrofti. Now we will discuss habitat. Wuchereria bancrofti inhibits in the lymphatic vessels, especially the lymph nodes of man and other vertebrates. MICROFILARIAE are found in the peripheral blood. These are also found in the chylous, urine, and hydrocele fluid. Morphology. Wuchereria bancrofti exists in three different morphological forms in humans, that is adult worm, MICROFILARIAE, which is also known as first stage larva, and third stage larva. First of all, we will discuss the morphological characters of adult worm. Adult worm look like a transparent hair-like nematode. It is creamy white in color. Its shape is filiform, which is tapering at both the ends. If we discuss the identification characters of male, it is 2.5 to 4 cm in length and 0.1 mm in thickness. Especially if we discuss the special identification features of male, its tail is curved ventrally and contain 2 spicules. Now we will discuss identification features of female. Female is larger in size as compared to the male. It is 8 to 10 cm in length and 0.2 to 0.3 mm in thickness. Tail end is narrow and abruptly pointed in case of female. Females are ovoviviparous, though they are liberating active embryos. Male and female coiled together and can be separated with difficulty. If we discuss the lifespan of organism, it is very long. It is around 5 to 10 years. Now we will discuss the first stage larva, that is MICROFILARIAE. MICROFILARIAE larva exists in the peripheral blood, often in the hydrocele fluid and chylous urine. Its size ranges from 244 to 296 micrometers in length and 7.5 to 10 micrometers in diameter. Its body is covered by the transparent hyaline sheath. They can live up to 70 days in human blood. Now we will discuss periodicity. Microfilarial periodicity is a special character of MICROFILARIAE. They become more active and more numerous in the peripheral blood when their intermediate host is more active. It has two periodicities, nocturnal and diurnal periodicity. When organisms are present in high number in peripheral blood at midnight, then it is known as nocturnal periodicity. When organisms are present in higher number during the daytime, then it is known as diurnal periodicity. Next we will discuss third stage larva. It is an infective form of filarial worm which is injected into the body of humans by its intermediate host. If we discuss its shape, it is elongated and filiform. Its size ranges from 1.4 to 2 micrometers in length and 18 to 23 micrometers in breadth. Life cycle of Wuchereria bancrofti. Life cycle of Wuchereria bancrofti starts when mosquito bite the humans for taking blood meal. When they are taking blood meal, they also inject the Wuchereria bancrofti third stage larva into the body of humans. From the blood vessels, Wuchereria bancrofti third stage larva moves towards the lymphatic vessels of humans. Within the lymphatic vessels of humans, male and female mate and produce sheathed microfilariae that migrates into the peripheral blood vessels of humans. When it is present within the peripheral blood vessels of humans, when another mosquito takes a blood meal from the humans, it also ingests the microfilariae of Wuchereria bancrofti with the blood meal of humans. Then the microfilariae moves into the midgut of mosquito, where it sheds its sheaths and penetrates into the mosquito's midgut. Within the mosquito's midgut, it further migrates toward the thoracic muscles of mosquito. Within the thoracic muscles of mosquito, it further changes into the third larval stage. Then the third larval stage further migrates into the mosquito's head. Within the mosquito's head, further it moves toward the proboscis of mosquito. From the proboscis of mosquito, it further enters into its next host when mosquito takes blood meal from the another person, which is the next host of Wuchereria bancrofti. Epidemiology. Wuchereria bancrofti is more prevalent in the warm and humid countries, especially tropical areas of the world like India, sea, Pacific Islands, Africa, South America, Central America. Pathogenesis. Wuchereria bancrofti causes lymphedema, which is abnormal accumulation of lymph in the tissues causing swellings of arms, breast or genitals. It is caused by the blocking of lymphatic vessels due to the accumulation or nesting of the Wuchereria bancrofti within the lymphatic vessels. Elephantiasis. It is the more advanced form of lymphedema, in which lymphedema is accompanied by the marked thickening of skin. Hydrocele. In this fluid-filled balloon-like enlargement of the sacs around the testes is seen. Diagnosis. There are various methods for diagnosing the Wuchereria bancrofti. First of all, we will discuss direct methods. Direct methods depend upon the visualization of larval stage microfilariae or adult worm. Microfilaria can be seen on the peripheral blood or other body fluids like urine, ascitic, hydrocele or lymph fluid. Organism can be observed by producing the wet smear examination under the microscope, where mobility of the organism can also be seen. Examination of stained peripheral blood smear, which is also useful in species identification, is based on the morphological features of organisms, especially the tail end. Thick or concentrated blood smears are used in case of very lesser number of microfilarias are present in the peripheral blood. Other direct methods which are used for the diagnosis of Wuchereria bancrofti are X-rays. Adult worms can be demonstrated in lymph node biopsy. Calcified adult worms are visualized with the help of X-rays. Doppler ultrasonography, also used for detecting the presence of motile adult worms in the dilated lymphatic vessels. Prevention and control of Wuchereria bancrofti. Insect repellents can be used when we are going outside to repel the insects, like its intermediate host Culex. Mosquito nets can be used to protect the humans from its intermediate host. Public education also plays an important role in prevention and control of mosquitoes. Thank you for watching.